We will go on, and we have a huge snow in Budapest at this moment. I could hardly make it to the hills, and apparently the same happened to our next speaker in Georgia. So we will move on, and we will continue with the presentation of Tamas Garay from Pázmány Péter University regarding exosomes. Tamas has been investigating tumor cell cultures and biological questions in cancer biology for 2 decades already. He is an excellent teacher and an excellent researcher, and we look very much forward to his presentation on his recent findings. Thank you.
Good afternoon, and thank you for the opportunity to speak here today. The title I got for this presentation is Hope or Hype. Of course, I won't be able to answer this question, but maybe I can tell you a few things that might help you to get closer to the answer. In my talk, after a short introduction, I would like to talk about 2 research projects that will hopefully allow us to get a glimpse towards the answer if extracellular vesicles are a hope or a hype in tumor biology. So first of all, the extracellular vesicle research is a quite new field in biology. Even the name extracellular vesicles was given only in 2011. But since then, there is an international society for extracellular vesicles. There are national societies like the HSEV for the Hungarian Society for Extracellular Vesicle Research, and there is a rising number of publications each year with more than 10,000 publications annually after the year 2020. So extracellular vesicles are lipid bilayer bounded particles. They have specific surface in terms of specific membrane proteins and also specific lipid composition. The classification is based on the MySEF guideline, the minimal information for studies of extracellular vesicles. It's basically based on the size of the extracellular vesicles. We are talking about small EVs and medium and large EVs and apoptotic bodies. The size and the composition sticks together with the origin of the vesicles. The small EVs are mainly derived from endosomal compartments, and they take mainly part in the intercellular communication. Medium-large EVs are often called microvesicles or exosomes. They bud directly from the plasma membrane and are mostly involved in controlling and remodeling of the extracellular matrix. And apoptotic bodies, as their name says, they are produced during the apoptosis. Almost all of the cells in the human body produces extracellular vesicles. As a biologist, I like to say almost. I have never heard an example from cells that were unable to produce extracellular vesicles, but there is always a chance. And despite this, we are able to find the extracellular vesicles in all biofluids, like urine, salvia, pleural fluid, and blood. They have a distinct role in tumorous malignancies. They are remodeling and taking part in the communication between tumor and stroma cells. They are part of the processes that forms the premetastatic niche, and they are also involved in genometastasis. If we would like to deal with EVs in an experimental setting, we have to take in consideration the difficulties we will have during our work. So the first of which is that EVs can derive from almost every cell. That means that they can derive also from red blood cells and platelets. So the plasma from the blood samples are to get isolated as fast as possible to avoid the excess number of red blood cell or platelet-derived EVs in the sample. This is often a technical issue during our work. The EVs overlap in size and density with lipoproteins, protein aggregates, antibody complexes and viruses. So the separation based on size or based on density, like filtering, like using a size exclusion chromatography, or like the idea to isolate them using centrifugation. A centrifugal force is have so these approaches always will have some inherent flaws. We won't have a completely clear population of extracellular vesicles. There are some projects about EVs where the idea is to isolate the EVs by binding them to a surface of beads or a surface of a isolating column with antibodies. But in this case, we make an a priori decision to accept only those particles to be EVs that have the given surface molecules, which is usually not proven. On the following slides, I would like to address two research projects, from which the first is about how to use microfluidic-based assessment of blood-derived EVs to address diagnostic questions. This project is in an early stage, but yet promising. So the first step during the sample preparation is to separate blood plasma that's usually collected and stored at minus 80 degrees. As mentioned earlier, the time elapsed between the blood sampling and the plasma isolation is critical. We would like to have it as short as possible to avoid platelet and red blood cell derived EVs contaminating our sample. The second step is to isolate the EV fractions using size exclusion chromatography. This is an easy method, and we know that we will have contamination of lipoproteins and protein aggregates in our samples. So in this case, it's probably better to talk about EV fraction or to talk about microsomal fraction instead of considering our sample as a pure EV isolated as pure EVs. For staining the samples, we use the fluorescent protein marker ETO 488. That is an amine-reactive generic protein marker. And the excess dye, we removed it and other with the second size exclusion chromatography step. The isolated EVs were subjected to NTA analysis and also to their evaluation using using microfluidics. We see the results of the NTA analysis. All the results were within the expected range of EVs, and we see no striking difference among the patient groups. That we couldn't differentiate with this method between the fractions of pancreatic cancer, prostate cancer, and colorectal cancer patients. If we go with the microfluidic evaluation, we need to add samples into the small channels, into the inlets of the microfluidic channel, in which we have two inlet sites for buffer and one for the sample. That results in a focusing of our sample into the central region of the channel. Where in the microfluidic systems, such forces such surface tension, capillary forces, and viscous forces move our sample to the side of the channel. So we lose the strict focusing which we have at the beginning of the channel. At the end of the channel, we have our sample apart from the center. As we use the fluorescent marker, we will have the fluorescent light basically dispersing in the cross section of the channel. In the next step, we assess the distribution of the fluorescent signal across the channel, and if we repeat the sampling in the different at the beginning, in the middle, and the end of the channel, in our experiment we used 10 physical points, 10 sections of the channel along the channel make these pictures, then the flattening of the signal intensity curves can clearly will be clearly visible after preprocessing of the signals. So we align them, we smoothen them, we cut them, and we did a background extension, and then we can see that we have a really centered signal at the beginning, and a much more flattened curve at the end of the channel. To do a mathematical and statistical evaluation, we need to fit a curve to a sample. So we fitted a two-sided sigmoid. So we fitted the sigmoid curve to the left and also to the right side of the samples, and we make this average. We determined parameters of this average sigmoid, like growth rate and amplitude, where the amplitude is basically the high of the of a sigmoid curve, and the growth rate is basically the slope of the curve. The amplitude showed a great variation among the samples, and which is quite straightforward in the sense that the amplitude is more prone to the technical issues, like the efficacy of the staining process, like the removing of the excess dye. But with the growth rate, we could we were able to distinguish between the three groups of patients. And if we put the two results together, the results got from the nanoparticle tracking analysis, where we could not get a difference between the patients groups, we can see with the microfluidic evaluation and with the growth rate as a parameter that there is a good visible difference between the three patients group, which makes us think that if we go further with this project and if we try to use machine learning algorithms to quantify the difference between the groups, then we will be able to differentiate between the different kind of cancers based on the extracellular vesicle, or based on the evaluation of the extracellular vesicle population from peripheral blood. The second project I would like to talk to you about is that is a cell-based and in vitro project where we use two cell lines from the same patients, one from the same melanoma patients. One, the pre-cell line was sampled before the treatment with vemurafenib, and the post-cell line was sampled after, or established after the treatment with vemurafenib. Both cell lines bear V600E mutations, and for both cell lines, we kept them, we cultured them, and we isolated the EVs from the supernatant of the cells, and using a two-step centrifugation, and followed by filtration before the ultracentrifugation step with 100,000 G for 1.5 hours. Then we got a microsome fraction that we used to subject EV characterization. That means we performed an NTA analysis, lipid and protein measurements, and we also checked for commonly used EV markers in the isolated microsome fraction. And in the other line, we also made a functional analysis where we treated the two cell lines each with both of the isolated EV populations. The results of the EV characterization revealed no difference between the EV fractions derived from the two different cell lines, meaning that we have no easily visible difference between the extracellular vesicle population produced by the cells. We evaluated the extracellular vesicles' effect on cell growth in a 2D model and also in a 3D, in a surrogate growth model, without seeing any significant or marked differences. We also evaluated the EV's effect on cell migration, as cell migration is a really important part in the process of metastasing. And so we we performed video microscopic evaluations. That means we basically made time-lapse videos from the cell cultures. We tracked the cells, and based on the XY coordinates of the cells, we calculated the migration parameter mean squared displacement. Where we could we can clearly see that there was an increase in migration in the pre cells after the treatment with the post cell derived EVs. To facilitate statistical comparisons, we calculated the difference in the area under the curve of treated and control cells migration. This is only a slide as a technical remark to show that we basically plotted the two curves on one graph, and then we can visualize the differences between the two area under the curves, and we could subtract these from each other, and so we got a change that is caused by the treatment. In this case, after this, we were able to make a statistical analysis for the migration parameter, and we found that the post-AV significantly increased the migration of the pre-cells. Just to remind us that we had the post cells that were isolated and established after the patient's treatment with vemurafenib. They were vemurafenib-resistant cells, and the pre-cells were vemurafenib-sensitive cells. So we asked us the question whether the migration-promoting effect of the post-cell-derived EVs is still there in the presence of vemurafenib. And after performing the experiments, we saw that the EVs derived from the post cells could increase the migration activity of the pre-cells, even in the presence of vemurafenib, so not only under control circumstances. So we first had to conclude that the EVs had a greater effect on cell migration than proliferation and sphere growth. Second, that the migration-promoting effect of resistant cell-derived EVs was not diminished by BRAF inhibitors. To get to kind of conclusion, then we can say that the analysis of EVs is still be useful to support diagnostic decisions, even if the task of purification and the separate analysis of individual EV populations is not fully solved. And we also can say that EVs play a part into the cell-to-cell transmission of certain features of the therapy-resistant phenotype. And that means that they could be potential targets for anti-tumor therapies. And on my last slide, I would like to thank all my collaborators without whom the research I just presented would not have been completed. And I also would like the opportunity to talk here today. And if there are any questions, I'm happy to answer them. Thank you.
Thank you, Tamas, for this nice presentation. I think it's very exciting, and we are not only trying to understand things from the patients, but also from basic and translational science. So I think these investigations are very important in means of that, and extracellular matrix and also extracellular vesicles, all that's outside of the tumor cells can be a fascinating environment. And our understanding, I think, until quite recently has completely missed the picture. So if we think that cancer is confined to a few cells in the body, probably we are wrong, and they are affecting other mechanisms and physiological processes alike. So thank you so much for your presentation. I will quickly look into the chat whether there is any question. If not, I thank you so much for your talk and being available there.
Thank you.
Hold on, there is one question.
Yep.
We will give the word to Professor Garay.
Yeah, okay. So when we are looking at live cell culture with high resolution phase contrast, we quite often see nanotubes which are broken off from the cells, migrating cells where lamellopodia actually get fragmented and sort of accumulating in the culture. So intuitively, we would think that if there are a lot of cellular movements with fragmentation of lamellopodia, that will show up. Do you consider those as part of the EV population? So mechanically broken lamellopodia, which just getting shedded off from the cell, does it show up in your fraction? Does it have a special name? I was looking for it in your list, but perhaps in the large EV fraction might be representing those.
So in the research aiming at EVs, we usually try to aim at the small EVs. So if some broken lamellopodia, I saw that I see them also on the wider microscopic in the wider microscopic experiments, but they should be removed in the size exclusion chromatography step, at least the majority of them. Actually, they are surely there in the cell cultures, and yeah, it's a good question to address them separately or together with the small extracellular vesicles.
And nanotubes, I mean, we time to time see nanotube formation between cells who after division they get separated, and many cancer cells love to produce nanotubes, and we managed to observe in pleomorphic adenomas actually using GFAP as a marker for it. But I wonder how often does it show up, and would that qualify also? Because nanotubes can also fragment it to tiny pieces. So they are in the...
Yeah, we did not measure them. So yeah, I see them sometimes. I see them not that often, and it's a good question. It might be interesting to look at them. So thank you for the idea.
Thank you.
Thank you.